This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. From the American Society for Microbiology and Microbe TV, this is TWiM, This Week in Microbiology, episode 144, recorded on January 19th, 2017. This episode of TWiM is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with free shipping by going to blueapron.com/TWiM. Hi, everybody. I'm Vincent Racaniello, and you're listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you?
I'm well, and you?
We have a wet San Diego, which is amazing. It's raining, and it's going to rain again. And our drought may be officially over.
Excellent. Yeah, well,
that's great for mushrooms.
That's unusual. That's unusual that you're getting rain. I'm glad to hear it. Yeah, we're getting a ton of rain here too, and it's unusual because there should be snow. Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello.
How have you been? Are you teaching this semester, or you have?
I taught last semester, and I'll be contributing to a graduate level course, and also the medical school infectious disease sequence later this spring.
Well, I taught my first virology class yesterday this semester.
Oh, good.
It's my class, so I always
Were they enthusiastic?
That. Hard to know, They're probably it's January, it's cold. They just came back from a month of spring break, so they probably don't want to be there. But I'll get them all riled up about viruses.
Especially after today's paper. It's amazing.
Amazing paper today, yep. Also joining us from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
How have you been?
I've been well, thank you.
Are you staying busy, Michael?
I'm staying busy. I'm like a one-arm paper hanger these days.
One-arm paper hanger. That's an
one-arm paper hanger.
What is that?
I learned how to wallpaper. I learned from my 2 uncles how to wallpaper, and they said the one thing you need to wallpaper properly is 2 good arms. And if you only had one, you can't do it because wallpaper falls on your head.
So doing something poorly is a one-armed wallpaper hanger? Is that
no, you just can't do it well. Because you have to use your head to hold the paper against the wall.
All right.
So you're very busy. you adapt.
Got it. All right. Well, we 2 weeks ago, our last episode, we announced a contest to win a copy of Manual of Clinical Microbiology. We have a winner we'll tell you about later, and I will do another book giveaway. I like it because people write and tell us a little bit about themselves, and that's always a lot of fun. Today we have a snippet and a paper for you, and Michael is going to start by telling us all about our laundry.
Yeah, it's always fun to think about everyday common occurrences, and this is a review paper that just came out in Applied Microbiology. It's a review by Dirk Bockmuehl, who's at the Rhein-Waal University of Applied Sciences in Cleve, Germany. And it's light on figures. There's only one, but this paper is heavy on facts. And the reason I selected this paper is I'm often asked to judge middle school science fair projects and sometimes high school science fair projects. And I often see a lot of projects that are looking at laundry detergents or soaps or all sorts of things. And I thought, this is a good time to look at the science associated with this. And, it's really good to see the practical side of microbiology. And to put this in perspective for the general audience, you don't actually think of laundry as a big business per se, but from a scientist's perspective, the animal antibiotic and antimicrobial market globally is expected to hit about $5 billion by the end of this decade. So it's going to be its annual expenditure, about $5 billion for animal antibiotics and antimicrobials. And, that's a big market because we use a lot of antibiotics in our food. But to put it in perspective, in the U.S. alone, we spend $3.3 billion on laundry, on laundry products.
Are we the highest spender in the world?
No, we're not the highest spender in the world because I couldn't find that statistic, so I don't actually know. But the Tide alone is $1 billion expenditure in the U.S., and it's absolutely fascinating just looking at this. And so our goal when we do laundry is mainly to remove stains and dirt from recently worn textiles. But more importantly, it's the elimination of microbial contamination. And this is nothing new to microbiologists. The transmissions of diseases via contaminated clothing is an old and established risk. The earliest documented spread of disease through contaminated clothing is found in Leviticus 13 to be precise, of which the Bible states that the garment that the plague of leprosy is in, whether it be woolen or linen garment, whether it be the wrap or woof, which is woven linen, whether in a skin or anything made of skin, shall be washed or burned according to the judgment of the priest. So this is we've been worrying about this for a long time, and I'm impressed that we sort of knew about the germ theory of disease, especially when it came to laundry, way back. The Old Testament is what, 4,000 years old, give or take.
Wonder aloud about something. Mm-hmm. Why a toga virus is called toga? Are they associated with Roman togas?
No, because when they were first visualized in the electron microscope, it looked like they had a toga on them.
Too bad.
Yeah.
Too bad.
But Michael, in the Bible passage you point out, that's for leprosy, which is characterized by open wounds on the skin that have the microbe right there replicating at the surface.
But they knew it was transmissible.
Right. But the concept that clothes in general can spread infectious disease is not really a general one. That's a very specific case, right?
No, that is a very specific case. But and that's where he brings us the data, and the data that he's going to share with us in his review article is really pretty compelling. And laundering is really designed to remove stains and dirt from our lightly worn textiles. And it's also to reduce the risk of this microbial contamination. And industrial and institutional laundering follows the Schaechter circle, which is dependent upon 4 principal criteria: the time that the garment is being interacted with, the temperature at which you're laundering it, and keep this in your head as we go through our discussion of the data. To convert centigrade to Fahrenheit, you multiply the temperature by 9/5 and add 32 to go from centigrade to Fahrenheit. And to go the other way from Fahrenheit to 32, you subtract the 32 and then you multiply by 5/9. Because he reports everything in centigrade. The curse of being an American, where I don't attribute centigrade to temperatures that my body understands, like how hot is the hot water in a shower. And finally, the mechanical action and the chemistry. And I learned a lot of really neat things reading this review about these processes. Each factor contributes or accounts for a certain percentage of the total cleaning performance. And in principle, you can compensate one for the other. You can add more time to a wash cycle. For example, if you recently shopped for a new washer, you've probably stumbled across the array of programs that our Internet of Things now has. And most notably, you'll see a lot of washers are now coming out with eco programs that principally rely on colder water temperatures in order to save energy. And in fact, the detergent manufacturers are changing their formulation so they're more effective at working at lower temperatures. And you've probably seen that as you've gone down the supermarket aisle looking at these things. So I had sort of intuitively understood this before I read Bachmuel's paper. As he took us through each of the sections, it began to make more sense to me, and he put all the references in one spot. So as I go off and review those middle school science projects about laundry detergents, which one is better, I'll at least be able to help help the young aspiring scientists learn how to think about this. The interesting thing is, as he takes us through the domestic laundering process, he points out that industrial and institutional processes are really very much standardized. They use extremely high temperatures. They typically use 60-degree water temperatures, and that's quite hot. 60-degree water temperature is
centigrade.
60-degree centigrade, which is extremely hot. It's 140 degrees, and of course that will scald your skin if you happen to step into a shower at 60 degrees centigrade or 140 degrees Fahrenheit. And the bleaching agents that we add to ensure sufficient hygienic conditions or hygienic reconditioning of the textiles, and many institutions do bleach these products in order to ensure that the viruses that may be carried along are indeed gone. But domestic laundering, the processes are less defined and not always led by purposeful aims. So he continues to point out throughout the review the goal that energy efficiency for household appliances is really what's been doing a lot of these things. And so consequently, our laundry that we're doing these days is not as clean as it once was, because the washing temperatures, at least he highlights in Europe during the last decades, and the convenience aspects led to an increased use of these liquid detergents that don't appreciably contain bleach, which in turn impacts the antimicrobial efficacy of domestic laundering. So in this snippet, we're going to talk about these four principal categories. So starting with temperature, in 1972, the average domestic hot water wash was 63 degrees, 144 degrees Fahrenheit. Today, the temperatures have been significantly reduced down to 46 degrees, which is 109 Fahrenheit. And so that's the typical temperature of the average American hot shower. 109 degrees Fahrenheit is the hot shower that most of us will jump into. And this reduction in temperature results in a reduction in your energy costs by 50%. So you drop from 144 degrees Fahrenheit down to 109, and you save half of your energy costs. So this is how they're making more energy efficient things. Hot is no longer the hot that my mother used when she taught me how to do laundry when I was a teenager. So from a microbial perspective, he points out that inactivation of norovirus or fungi, principally athlete's food, can only be achieved at temperatures greater than 104 degrees Fahrenheit. And a lot of us do a lot of our laundry at cold water temperatures where they're not even heated at all. And so this is a significant point for me, considering how many of our children acquire norovirus. And even after recovery from the diarrhea phase of norovirus, they continue to shed the virus into their undergarments, and they drop it everywhere. And that's why norovirus is so communicable. So, if you think about if your kid has had a recent bout of diarrhea and you don't want it to go through the rest of your house, you may want to consider laundering their undergarments with very hot water over 100.
Wait. I read this fairly quickly, but I don't believe there's any evidence for people getting an infectious disease from their clothing.
No, there is for Shigella.
There's a huge dilution effects.
This is correct, but norovirus, actually, you only need 2 to 20 in order to get the virus.
But
and you're
would
typically
you have to suck on the clothes in order to?
No, you just have to pull them on, and they're on your hands, and so you can inoculate yourself.
Put your hand in your mouth.
And those studies haven't been done, but norovirus is extremely communicable. So as I was reading this, I'm trying to think about, is bleach worth it? Is high temperatures worth it? Because I was like you, Michele, going, I don't think I have seen the literature pointing this out. And so what industry has done in order to deal with this is they've extended the wash cycle. And again, this gets back to Michele's comment a moment ago about the dilution effect, because the measurement has been the removal in laundering, it's all about the removal of stains rather than the removal of microbes. So the bulk of his review is focused on these factors that influence the hygiene efficacy of the laundering process. And there's only one figure in the paper, and he effectively takes us through that figure as he is thinking about where the microbes come from. And so the majority of them come from wearing the garments. They principally are from our skin flora and the excretions. And then as you go through this Schaechter cycle, if you
surprise.
Yeah, and many of those are good microbes. You want them on your skin.
You want them on your skin. But the second phase is you get cross-contamination of biofilms. Now, his review doesn't specifically address this, but in the United States, we have seen what has gone on in Europe. We have seen the popularity rise of the front-loading washers. And the front-loading washers have begun to get problems with biofilms growing on those gaskets. And that cross-contamination is becoming a problem of the biofilms actually that are recalcitrant to some of the soaps that are being used. They're persistent in the environment, and of course the presence of persister cells that we learned about in the last paper that we talked about.
But norovirus, the example that you use, something highly contagious, is not going to be affected by a biofilm.
No.
So I'm still having difficulty seeing the connection between using lower temperatures and spreading infectious disease.
Okay, so let's jump to the data. So Weiskel and colleagues, he points out, observed a log reduction increase of approximately 2.5 log units when the temperature was shifted from 24 degrees centigrade or 75 degrees Fahrenheit to 68 degrees centigrade or 154 degrees Fahrenheit. Again, putting this into perspective from the hot shower approach, you should not go about 120 degrees Fahrenheit, for this can cause the scalding burns. And so what Weiskel and colleagues showed is that as you increase temperature, you increase the ability of the temperature in the laundering process to inactivate or kill microorganisms. And another study done by Schillinger in 1975, he learned that the log reduction increased by 3 orders of magnitude simply by moving the temperature profile from 38, which is 100 degrees Fahrenheit, to 49, which is that scalding shower. And so studies done in this century, if you wish to reduce the microbial concentration, a temperature of 50 degrees centigrade is an ideal temperature. And most of the tankless hot water systems that are being deployed across the United States have their set point of those hot water tankless systems to be set only as 120 degrees as their max. So you're often hard pressed to even get the hot that is anticipated. I was talking with a colleague of mine who has spent some time in Germany, and he was talking about the German coin-operated washers, and they have a setting called Koch-Buttwasch, which means it's a cook/colored wash is the literal translation. And he says that hot is so hot, he ended up shrinking his clothes. So our definition of hot in the United States is not the same as what hot in Germany is, at least for coin-operated front-loading laundry machines. Next up, he asked us to consider chemistry of the laundering process, and he broke this into subcategories, which were fascinating. There were 3 major groups. One were the surfactants. The second one was the bleaching agents, and the third one was the Quaternary ammonia compounds. And I'm well familiar with quats because we use them routinely in the hospital to disinfect inanimate surfaces and to inactivate microorganisms. But quaternary ammonium compounds, because they're obviously positively charged, don't work all that well if you happen to have a anionic detergent present. So they have to be added after you've effectively removed the stains and the odor part. So the surfactants, which my mother informed me when I was a teenager was soap, this is the most important component in the detergent as it accounts for the basic cleaning of the garments, and it's designed to remove these hydrophobic soils. So you're effectively doing solution-based chemistry. The scientific literature is slim on this, but Bachmel cites a paper by Hanisch that investigated the laundering process with and without detergent. And Hanisch found literally no effect when comparing the microbial load on artificially contaminated swatches. The only one he really saw a difference was with the Staph aureus and the Enterococcus hyrae, which exhibited a considerably higher log reduction rate when washed with a detergent compared to water alone. And in fact, when they did the, if you will, in vitro experiment doing a suspension test, they saw no antimicrobial effects attributed to the detergent. And this was in a paper by Bachmel and his other colleagues that was published in 2016. Bleach, the section on bleaches was really fascinating to me because I hadn't known too much about German laundry products. And here in the U.S., when you say bleach, the branding specialists have taught us to say Clorox, which is sodium hypochlorite, which is typically at a concentration of about 6.5%. And that's traditionally used in Southern Europe and here in the U.S. and Germany. And Northern Europe, they principally use activated oxygen bleaches, which are principally peracetic acid derived from perborate or percarbonate and a bleach activator. And these are powders. And the bleach activator is tetraacetylene ethylene diamine, or TAD. And the advantage here is it's built into the detergent and provides both the whitening and cleaning as well as disinfection at lower temperatures. And he points out that when you add bleach or have these activated oxygen bleaching agents, it's hard to tell the contribution to the antimicrobial aspects.
So, Michael, I was taught you don't put bleach in color loads, right? Otherwise, you just put it in the lights. I mean, so that would say your colored laundry may have a higher contamination level, right?
Right. And traditional chlorine bleach is still the best, and I still add that to my whites because it increases the antimicrobial efficiency of the process by 3 to 4 orders of magnitude, and I can get away using cold water temperatures. The thing that I didn't know when I read through this paper is the discussion on the quaternary ammonia compounds, which are principally benzalkylinium chloride, or BAC, or DDAC, which is didecylamethylammonium chloride. And these cationic agents are in direct conflict with the anionic agents, which are the detergents. And so they typically sneak them in with the rinse or with the fabric softeners. And the quats attach themselves to the textiles and the polymers, as those traditionally are negatively charged. And we've all had the static discharge in the wintertime when we're trying to separate socks. And it's interesting that the fabric softeners are also cationic based. So they stick to the fabric and they add loft to it, and they effectively are responsible for making them feel soft to our touch. He then takes us through the mechanical action and time, and there weren't a lot of data here. And again, temperature is the controlling element here, and it was the most help at eliminating microbes. And there have been papers in 2016, Brand and colleagues performed a suspension test and compared the data with the results obtained from a washing machine. And it was found that microbes were reduced on textile surfaces during laundering at 20 or 68 degrees Fahrenheit to 40 degrees or 104. And they report on the various things that they have tested. But Bachmel continues his review by, again, calling into perspective this hygienic efficiency beyond the Sinner cycle, there are many other variables to consider. And
so Michael,
materials.
How do you square this with the discussion about the hygiene hypothesis and the missing microbes, and that we're actually, by using antimicrobial products on our hands, and that we're actually increasing other health problems? I'm just not convinced our laundry needs to be sterile. A lot of microbes are harmless, and many are good.
I know. And which is why I thought this would be a good discussion to have, because it would engender a discussion. And the hygiene hypothesis is certainly very important. I mean, the argument is we're too clean, and that's principally driving all the autoimmune conditions that we're seeing develop in ourselves as well as our children.
And allergy, asthma,
all of these things. But again, this was the first comprehensive review that I've seen in a while that is, again, taking to task this whole problem of how to do the science.
Well,
he does bring us to the European standards that are talking about how to measure the efficacy. And the paper is open access, so I'll commend that to the listeners. And he ends up his review by asking us, will inefficient laundering result in harm or an adverse effect? And this gets to Michele's point, is whether or not our aggressive nature of adding bleaches and really scrubbing the life, if you will, out of things is really going to cause harm. And for the most part, most of us attribute whether laundry is clean as the absence of odor. And a lot of laundry products actually put in perfumes. And those of us who are sensitive to various soaps, of course, are buying the de-scented materials to effectively do that. And the other thing that he points out is the sweat ingredients may serve as a nutrient for remaining bacteria that are able to grow and metabolize the sweat compounds if the humidity is high enough. But again, it all goes back to what we want in terms of clean. I think from a hospital perspective, we want our sheets and pillowcases and towels to be very clean. And in sports complexes, I think the NFL has established that towels were indeed the fomite, even though they were cleaned. The absence of bleach in the process in the NFL resulted in those towels indeed transmitting superbugs amongst the players that had open wounds as a consequence. And the net result was you ended up transferring MRSA from person to person in that particular process. But again, they were wounded because they were playing football and they had abrasions, scrapes, and whatnot. And he concludes his review by introducing us to a reference about microbial risk assessment, which I think is the principal take-home message. And this is the quantitative microbial risk assessment. And this is a print that just went up live before the editors were done typesetting it and whatnot. And it's missing the reference by Gibson, who calculated that the probability of acquiring a Shigella infection via contact with contaminated clothing could be reduced by 90% through laundering. And Shigella is enteric pathogen that is transmitted fecal-orally. And the data, and I went and looked at the Gibson paper, is pretty convincing. And while the reference is not in the manuscript because it hasn't been proofed yet, I found the reference, and I'll put it in the show notes if you're interested to learn about using quantitative microbial risk assessment for the evaluation of the benefits of laundry sanitation. And that was in the American Journal of Infection Control in 1999.
So Michael,
overall...
Go ahead. Let him finish. I got things to say. Go. You can finish up. Go ahead.
Overall, I think the way my mother taught me to do laundry is probably still valid. Use bleach for the white clothing, and that's principally, I wear white socks when I exercise, so that'll kill off any of those damn little fungi I'll pick up in the gym on my feet, so that'll prevent the athlete's foot from running around if I bring it home. And it'll take care of anything that I may accidentally shed on my undergarments. So overall, it was an interesting review to read simply because I didn't know much about the German bleaching process and the Kochen Bootwash, which is the coin-operated where they cook their clothing. And according to what I could find, it appears that those wash machines come out at about 60 to 70 degrees centigrade.
Elio, you're going to say something?
Oh, not really. I think this is an illustration of how difficult sort of real-life situations can be.
Yeah.
I mean, there's the data tend to be confusing at times. And I think it's typical of this kind of a study. But we'll hear more about it. I'm sure that people are going to do much more, and we'll come up with some conclusion in time. But, you know, it's a little bit like nutrition studies. They're intrinsically so darn difficult that, you know, it's not surprising that every month you hear something contradictory. So it's that kind of a stuff. I'm glad somebody's doing it, but it's going to take a lot of work to sort it out. But thanks, Michael. I appreciate it.
Let me just add a couple of comments that I've been thinking about. First of all, when I was a kid, my mother used to boil the diapers because we didn't have disposable diapers. Oh, yeah. Right? So she'd rinse them out.
My mother did too.
Rinse them out.
My mother did too.
Boil them and then wash them. So that's important because you don't want to be giving fecal pathogens to the kid. We've always used scent-free laundry detergent. And I think at home, the main point of washing is so you don't have odoriferous clothes. Many of us know people who wear clothes for weeks and weeks and they smell. And that's not good. But you can imagine in hospitals, as Michael said, but hotels, you want your towels and sheets to be really clean, right? And I'm sure they use industrial high temperature launderers to do that.
Which is why most towels in hotels are white.
Yeah. I guess they add bleach, right? And they put them in high temp.
Bleach.
And they probably wear out quickly and they go through them, right?
And the dryer is also going to kill a lot of microbes.
Yeah.
Well, that was the one thing that was absent from the discussion that I was going to bring up, but I saw it was going long, and that they don't consider the drying activity.
Yeah.
And maybe that's because in Europe, a lot of people hang their clothes out on a line to dry, whereas in the U.S., the majority of us actually throw them into a high-temperature dryer.
Yeah, and there you have the natural UV killing the microbes.
Yeah. You know, we recently got a brand new washing machine. The old one broke. And this one has drawers where you put the soap in. So there's a drawer for soap. There's a drawer for fabric softener. And then there's a new one for OxyClean, right?
Yeah.
Which is sodium percarbonate, hydrogen
it's that
peroxide.
Activated oxygen bleach.
Yeah.
Yes.
So basically you can use that for anything and it, removes stains and so forth. So I don't know what that contributes to sterilization as well, but it must.
Or what it contributes to our water supply when it goes down the drain.
Well, these new high efficiency washers, they don't fill up the thing with water and wash anymore. Right. They just spray little bits of water. I don't even know how it works because it seems to me that you can't possibly. But
do you smell good, Vincent?
Yeah, I smell fine. Nobody objects, so that works.
No one's complaining on the subway.
It's working. Thank you, Michael. That's very interesting.
Thank you.
I bet our listeners will have some thoughts about that. This episode is brought to you by Blue Apron. Blue Apron's mission is to make incredible home cooking accessible to everyone while supporting a more sustainable food system and setting the highest standards for ingredients and finally building a community of home chefs. Blue Apron delivers seasonal recipes along with fresh, high-quality ingredients to make delicious home-cooked meals. Every meal comes with a step-by-step, easy-to-follow recipe card and pre-portioned ingredients. You can do them all in 40 minutes or less. If you spend a lot eating out or at high-end grocery chains, you can now spend under $10 per person for healthy home-cooked meals. I have to say, I love to eat. I'm not a fan of cooking because I don't like getting all the stuff together. And I love Blue Apron. I tried it out last week. Man, it's really easy. They give you everything you need except salt, pepper, and oil. All the spices that you'd need. You'd have to go out and buy a whole bottle of them. They send them to you, and they're fresh. The meats are fresh. The fishes are fresh. Man, I just love it. I thought our listeners would really like this too. I approve what ads we do, of course, and Curiosity Stream was, of course, obvious for Twix listeners, but this one I like because I think everyone likes to eat. Not everyone likes to cook, and you can save money. You're not going to gain weight because it's a fixed amount of food, not a huge amount, and you can only eat that and nothing more. And it's fun to cook. Now, Blue Apron sets the highest quality standards for their community of over 150 local farms, fisheries, and ranchers across the United States. For example, seafood is sourced under standards developed in partnership with the Monterey Bay Aquarium Seafood Watch for sustainability. Beef, chicken, and pork come from responsibly raised animals, and produce is sourced from farms that practice regenerative farming. And this is what I like, because they ship the exact amount of each ingredient required for a recipe, they reduce food waste. You can customize your recipes every week depending on your dietary preferences. You can choose a delivery option that fits your needs. They're very flexible. No weekly commitment. You only get deliveries when you want them. And tonight, I'm expecting a box to be delivered at home, and I can't wait to get it and start cooking. I don't tell them what to send me. I let them pick, so I'm surprised. It's really cool. They deliver, by the way, to 99% of the continental U.S. You can choose from a variety of new recipes every week, or you can let Blue Apron's culinary team surprise you, which is what I like to do. Last week, we had chicken souvlaki with roasted broccoli. Oh, it was so good. There's more to it, but I can't remember. And we had crispy salmon with Meyer lemon aioli. And each meal comes with side dishes. For example, here's some examples. By the way, they never repeat a recipe in a year, so you're not going to get bored. Can you imagine? Every week you can have something different. Spicy shrimp and Korean rice cakes with cabbage and furikake. Pork chops and garlic piccata with scallion rice and spinach. Cashew chicken stir fry with tango mandarins and jasmine rice. You can see there's always a main dish and a couple of sides. They make my mouth water. I shouldn't record this so late because I'm hungry. Check out this week's menu and get your first 3 meals free with free shipping on top of it by going to blueapron.com/TWiM. That's blueapron.com/TWiM. You will love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. So don't wait. This is a really good deal. Blue Apron, a better way to cook. All right, I want to tell you about a really interesting paper that was just published in Science magazine as a report. It's called Assembly of a Nucleus-Like Structure During Viral Replication in Bacteria. And the authors are Schaechter, sorry if I messed that up, Nien, Kana, Brillo, Herb, Coker, Vavilina, Newton, Buschauer, Poliano, Villa, Agard, and Poliano. Kit and Joe Poliano. It's out of their laboratory at UCSD. And, I visited their lab a long time ago on TWiM 115, profiling the Polianos. Went right into their lab with video and talked about their work. And part of it was a piece of this story, and that is they're working on a bacteriophage of Pseudomonas called 201Phe2-1.
It's a strange Pseudomonas, by the way. It's not a Rugosa or one of the regular ones.
Pseudomonas chlorophyll. So, Elio, is that something people work on in the lab because it's safe?
I don't know.
I don't know. Okay. Anyway, they work on a phage of this, and they previously showed that the phage genome encodes a tubulin-like cytoskeletal protein called PHUZ, P-H-U-Z, and this helps to position replicating phage DNA at the middle of the cell. And they talked a little bit about that on that TWiM 115. And what they do in this paper is they're really interested in this positioning of the replicating phage DNA by the tubulin. They want to know what other proteins are involved in this. So they did a really big study where they used mass spec to identify proteins in infected cells. They use mass spectroscopy to identify proteins that are turned up in infected cells, including phage and as well as host protein. And they take some of these, they fuse them to the gene encoding green fluorescent protein with the idea of localizing them in both infected and uninfected cells. If you go back to that TWiM 115 and many of our other discussions of work from the Poliano lab, they're a very visually oriented laboratory. They like to use microscopy, time-lapse, and static microscopy to visualize what happens in cells, and that's what they're doing here. So they find there's a particular phage protein called GP105. It's the most highly produced protein in infected cells. Now, you can take the gene for GP105, produce it in uninfected cells, and it's a diffuse protein throughout the bacterial cytosol. But when you infect them, all of a sudden this protein redistributes and forms a spherical structure that surrounds the phage DNA. This happens at about 45 minutes after infection. And they find this in every infected cell. Most of the cells have one of these spherical structures. Just a few of them have 2 or 3, and it's very stable.
And you may also say that this structure was unknown to science previously.
Yeah, this is really an amazing thing, and maybe later we can get back to that. No one's ever seen this before, and it may be because of the way they're looking at it. They're taking individual proteins and tagging them with a fluorescent protein, GFP, and then you can see them and what goes on during infection. It's not easy to do otherwise. So this is a very stable structure. If they photo bleach it, in other words, remove the fluorescence and then wait for it to return, it doesn't recover. So all the proteins are there and they're staying there. So they did some more experiments, a lot of experiments. It's a very visual paper. I don't know, this is behind a paywall maybe, but those of you who can see it, there's some movies available. There are lots of gorgeous photographs.
I mean, I commend this to any reader that has access to this journal because it's really spectacular visual stuff.
Just exquisite. I did post in the show notes a YouTube video that people will be able to watch without a paywall.
Yeah, they have, the paper is relatively easy to read because it's mostly a visual paper. There are a lot of figures to look at, and they're explained very well. So what they did next is some time-lapse microscopy to figure out the timing of the formation of this structure. So they produce the protein in cells, then they infect, and then at different times after infection, they do their microscopy. And so the GP105, the component of these compartments or spherical structures, is green, and then they make Phage, which is the tubulin-like protein, they make that red. And what happens is, this is really amazing. First, earlier infection, you see a small focus at the cell pole. It's green, so these little spherical structures first form at a pole of the cell, and then there are red filaments at both cell poles. With time, the red filaments push the green spherical structures towards the center of the cell. They push it towards the center until it gets to the center of the cell.
Yeah, when in the movie you can almost feel the central structure, the phage structure, saying, "Stop shoving already."
Yes, it's very funny. And as these structures are moving, the shell increases its size. It reaches mid-cell, and then DNA replication begins. Phage DNA happens within this shell. The shell grows inside. And once the shells reach the center, they oscillate back and forth because they're opposing filaments of this Phage protein, which is a spindle, and it pushes one way and the other. And if a particular sphere happens to have spindles pushing it on opposite sides, it rotates. So you can see movies of these things rotating, which is really
emphasizes that there's force being applied to that shell.
Absolutely. And they can also examine a mutant where the gene for Phage has been altered, so the protein cannot hydrolyze GTP, and that's, of course, needed. That the energy from that would be needed for the movement of these filaments.
Yeah, but the rotation of the structure, the nucleus-like structure, has no explanation really. I mean, is it a passive thing? Does it matter? Is it important? We don't know. So the nice part of this paper is that it presents so many new questions.
A lot of questions here. Yeah, I mean, the whole movement, we don't understand why, it should be in the middle of the cell and so forth. So these are interesting questions. But they do use this mutant where the GTP can't be hydrolyzed and movement, the shell remains at the cell pole. It doesn't move. Now, an interesting question, they didn't really address this. I just wonder, in a virus-infected cell with a Phage that cannot hydrolyze GTP, would you go through the whole viral replication cycle? Right?
Yeah, and what's the yield? Do you get less phage from that?
Exactly. So they don't answer that. It would be an interesting question to know if you need to get in the center of the cell or can you be at the tip and so forth. Then they looked at 52 different phage proteins in the infected cell. And each of these is labeled with GFP or red, and they can
That's quite a, by the way.
It's amazing. Oh, it's a huge amount of work.
Amazing stuff, yeah.
And they say, they want to know which are in the shell and which are excluded. So some proteins get in and some are not. So there's an interesting exclusion here going on where some proteins are let in and some proteins are not. For example, proteins involved in DNA replication and transcription, the production of mRNA, like a phage DNA helicase, a phage ligase, RNase H, Rec A, RNA polymerase subunits. These are all in these spheres, these compartments. Also, a host DNA topoisomerase 1 protein gets in. Of course, all these things are needed for DNA replication and transcription, and they co-localize with DNA in the shell. On the other hand, proteins of the host involved in translation, like ribosomal proteins, initiation factors, release factors, they're outside the shell. They're in the cytoplasm.
And the pictures are so convincing. It's really beautiful.
But the big mystery is the nature of this membrane. They really, at this point, know very little about it, right?
It looks like a protein.
Is a membrane.
Yeah, it looks like a protein shell, right? And they say that it's very sharp.
It does not look like a lipid membrane.
Exactly. It doesn't. It looks very different. It's very sharp. And they say when they stain it, it looks like all GP105. So it looks like a protein shell, which is really interesting.
But it's worth pointing out that eukaryotic nuclei also have a nuclear lamins proteins that make a shell and give them structure. So.
And enzymes, phage enzymes that are involved in nucleotide synthesis, for example, thymidylate kinase, these are also excluded from the shell. So they say this compartmentalization is very much like that of the eukaryotic cell nucleus. That's why in the title they call it a nucleus-like structure. Now, when proteins are made, they do experiments, time-lapse, microscopy. They'll produce a protein labeled with GFP. For example, they do this for Rec-A, the phage Rec-A homolog, which is involved in DNA replication. They make it in the cytoplasm, then they infect, these structures form, and then the proteins are brought in. And for Rec-A, nearly all the protein is brought in. And so basically these proteins are brought in post-translationally. They also show that when the shell rotates, everything inside rotates as well. The other thing they did is to use proteins to study phage assembly. So how does this relate to phage assembly? So there's a major capsid structural protein called GP200. They label it with GFP, and then they do an infection. And at 40 minutes post-infection, they can see empty capsids assembling near the bacterial membrane. All right. So
cool.
And then these empty capsids migrate to the phage compartment. They sit on the outside. They rotate with the compartment. So they must be docked onto it pretty tightly. And when they stain it with a dye for DNA, they can see DNA entering the capsid as it's docked at the surface of this compartment.
Amazing.
It's just amazing.
Watching
it is.
Morphogenesis of phage.
When I looked at that, I was wondering if the phage did this to figure out how to dewater the DNA, because that's one of the hallmarks of phage is that the DNA is effectively dry, and that's what makes, really supports the stability of the DNA in that phage particle so it can go out and do what it needs to do.
Yeah, maybe. They also use cryo-electron tomography to visualize this compartment. So this is a technique where, you do electron microscopy on frozen specimens, and you tilt the specimen stage and get pictures at different angles, and then you can assemble them in 3 dimensions with software, very much like a CAT scan. And what they find is this compartment has a sharp border. It's very different from a cell membrane, as we've said, and that they say that makes sense because it's mainly composed of a protein, GP105. It's got a thickness of 5 nanometers, completely enclosed. There are no holes, and it excludes GFP. If you just produce GFP in the bacterial cell, it doesn't get in to the center of this compartment. So it's a barrier to diffusion.
One technical point. If you do, these bugs are too thick to do regular cryo-electron tomography. You don't see anything.
Yeah, right.
So they use a technique developed by one of the co-authors, Villa, which is called cryo-focused ion beam milling, which essentially is to take the cell, which is frozen, and mill away part of it. So it's, you're sort of removing it as if you would in a wood shop with a plane. You remove layers of it and you make it thin enough so you can do the cryotomography. So this is
right.
It's like slicing ham at the deli.
That's right. Exactly right. And they don't see any ribosomes inside these compartments when they do this cryo-electron tomography, which is consistent with their localization results before. They can see capsids in the bacteria at the bacterial inner membrane. These are empty. They can see some docked on the compartment. Some of them are empty. Some of them have various amounts of DNA. Some of them have a connecting collar that will eventually be attached to the tail. And then eventually when they're filled with DNA, they're released and the tail is assembled in the cytoplasm. So away from the compartment, they see filled capsids and also complete particles. So the idea is that the phages acquire their DNA at this compartment. They move off, mature in the cytoplasm, and of course, they're released as the cells lyse. So basically, that is the story. This is a nucleus-like structure. It's assembled in phage-infected cells. The DNA replicates in it, and it's positioned there in the center of the cell by a tubulin-based spindle. They believe that there are a lot of questions here. First of all, the phage mRNAs are probably produced in the compartment because the transcriptional machinery is there. How do you get out? How do things get in? How is there selective transport? These are all very interesting questions. Do other phages do this? Why haven't we seen it before? It would be interesting if this is a universal property, right?
I have a feeling that phage people don't usually use the microscope enough, so that it may be that some other phages do this, but they've never been seen because nobody's looked.
Right.
Well, and it's not just looking, right? You have to go through the work of making functional GFP fusions.
Yeah, maybe it's not been
There's
done.
a lot of cloning in this paper.
Right.
A lot of cloning.
But you could see, just staining, you could just DAPI staining, you could see something in the center of the cell if you looked. So, you don't have to go through the whole thing to discover phages that are candidates for this. It could be
we should also emphasize they chose a very large phage. So this one has a genome of 316 kilobases. So that makes it easier to spot and track this morphogenesis program.
So
of course, to me, the big question is why?
Yes, that's right.
Why does this have the magnificent device? Why does it happen? What good is it?
So one of their suggestions is that it's a way to shield the viral DNA from host, defensive mechanisms. Which
Restriction ends and nucleases.
Exactly.
Yeah.
Maybe, CRISPR.
Well, there's more to it.
CRISPR-Cas9.
What fascinated me is the implications for evolution. We may be seeing how prokaryotes gave way to eukaryotes.
Exactly. I said the same thing. I said here in my notes, does this suggest that the nucleus is a viral invention?
Yeah.
That's what I think.
Right.
So
now, I can tell you that the authors try to bring that into the paper, but the editors nixed it.
I'm sure, because there's no mention. This is the problem with science. The papers are so short, you can't speculate. And that's what science is about at the end. You should be allowed to speculate. Yeah,
they should have been allowed to add a sentence or two at the very end.
Yeah,
say the implications are
other people like us are going to do it for them.
Well, no, that's the intent of the podcast.
That's right. Exactly
we can
right.
interject.
So here's my view of viruses. Here's one hypothesis out there is that in the early years of Earth, after it cooled down enough and there was an organic soup, that there were self-replicating nucleic acids, which we view as precursors of viruses. And then maybe some cells formed at some point. It's a big leap, but that happened. And these maybe early cells didn't have nuclei. Maybe they had RNA genomes because these self-replicating genomes were RNA-based. Early on, again, there were no proteins. It was just nucleic acid that self-replicated.
So here's
ribozymes.
a title for the show.
Yeah.
Did eukaryotes invent anything?
Yes, right.
Laundry.
That's
We
right.
invented laundry.
They didn't invent mitosis, it looks like.
No. This could be mitosis.
And so then the viruses decided to enter these cells that formed and replicate, and maybe they figured out that a nucleus would be a good idea, and so that the cells then stole them. And so that's actually an idea that cells arose from viruses, and then the viruses, we might as well replicate in these cells because they're a nice place to replicate, and maybe they gave them the nucleus as well as everything else. So I find that really interesting.
I do too.
I thought this was a spectacular piece of work.
So obviously, Elio, since you're in touch with these individuals, you knew
Yes.
this was happening, right?
Oh, yeah. Yeah, I saw it happen, and I was cheering on the sidelines.
Good. Yeah, it's a great story. And
I was able to communicate with the lead author, Vorapin Chekuratsika. Sorry. who told me a bit about his background. So he's from Thailand. He was a biochemistry major in college and was then fortunate to be awarded the Royal Golden Jubilee PhD scholarship at the Chulalongkorn University in Thailand, which covered his entire tuition for the PhD and included up to 1 year of conducting research abroad. So he took his year abroad at UC Davis, working in the lab of Peter Armstrong, and got to work very closely with the PI, which got him quite excited about research. So after his PhD, he was a postdoc at UCSD working on arteriosclerosis at the School of Pharmacy. But as many listeners will be familiar, the funding ended in that lab, and he actually had to go find another postdoc position. So this was a real difficult period in his life. He applied to many places, but was eventually introduced by his roommate to Joe Pagliano. His roommate was already in the lab, and so this was his entrance to microbial cell biology that he did here. He said that many people contributed to this work, and it would not have been a success without it. So the Pagliano's lab and also the Vila lab contributed, as well as the Agard lab, who does the tomography. So he wanted to point out...
UC San Francisco.
At UC San Francisco, correct. So he wanted to point out that Katrina, a graduate student, was an expert cloner, and she was, thanks to her talent, they were able to make more than 50 of these GFP constructs within a very short period of time. And then...
That's not trivial.
No, not at all. And then another graduate student, Kanika and Alex, a postdoc, were the experts on the tomography. So all of those high-resolution images they were able to get in about 3 months. And then he really credits the leadership of Joe and Kit, two of the other faculty who were cheering this project on. He also pointed out that during rough times, he had a couple office mates at the lab, Elizabeth Montano and Hannah Tsunemoto, who cheered him on and kept him going. So I think this really emphasizes that although the general public tends to think of scientists as working kind of in isolation, quietly off in a corner. In fact, it's very, very social, requires a lot of people working together, talking together, and encouraging each other. And he really emphasized how important that was. He also said that Joe Pagliano is the best boss he's ever had. Joe is energetic. He has fun thinking about the projects. He's excited to know what's going on. Often he'll come in while he's sitting at the microscope collecting data for the first time, watching these GFP fusions, and just say, how's it going? Sometimes Joe will want to have a look at the microscope himself. So Joe happened to walk in while he was looking at the fusee, the microtubule filaments, pushing that phage compartment to the midcell. So they were able to share that excitement together. And when Joe first saw it, he literally threw his hands in the air and jumped and high-fived him and kept saying, "Isn't this fun? Isn't this fun?" So he tells us that this was one of the best days in the lab he's ever had. So his future plans are to return to Thailand. He'll be a professor at a university in Bangkok, where he hopes to develop the graduate education system there. He says that will give him an opportunity to be closer to family, which is a priority, but he also has always hoped to work in the United States. So probably he'll keep collaborations going and continue to work closely with his colleagues. As for advice for junior colleagues, he says, just remember life is not a bed of roses. There will be times when you might fall, you might get lost, but you've got to keep fighting through the bad days, keep pursuing your dream, and sooner or later it will be yours one day. And he certainly has demonstrated that with this beautiful paper. So congratulations to Vorup and all of his colleagues at UCSD.
Very good. Thank you, Michele.
Lovely.
The winner of the Manual of Clinical Microbiology, that's the 19th emailer, is Mitch. So Mitch is going to get the 2-volume set of this book. It's the 11th edition, published by ASM Press. Now, I have another one for you. If you'd like to win a copy of the second edition of the Color Atlas of Medical Bacteriology. This is also an ASM Press book. It is full of lovely pictures of plates of bacteria, gram stains, colony morphology, biochemical reactions, everything you do in the clinical lab, including antimicrobial susceptibility testing. Each chapter covers different bacteria, Legionella, Pasteurella, Clostridium, Helicobacter, etc., etc. Lovely photographs. If you are the 12th emailer with a subject line Color Atlas, you'll get a copy of this book. Now, why do I do this? Well,
why do you do this?
He shamelessly was looking for email.
I'm looking for email, basically. I get a lot of these books from ASM Press, and I'd like to share them with our listeners. I think a lot of them would learn from them. And I love hearing from listeners. Here are some of the examples of some of our listeners. Now, Mitch, who was the winner, didn't write anything, which is fine. But Kayla, for example, hey there, TWiM team. I'm a veterinary microbiologist in Cork, in Ireland. I'm a longtime listener of all the TWiX series, and I love every one of them. I work in a diagnostic lab in Cork, and the only thing that gets me through the day is your podcasts and buckets of tea.
Buckets of tea.
Thanks for the wonderful podcast. That's lovely. Justin writes, hello, TWiM crew. I listen to all the TWiX family, but TWiM happens to be my favorite, closely followed by TWiVo. Here's hoping I'm number 19. Hope Yins, Y-I-N-Z, which is Pittsburgh, plural of you. Have a fantastic 2017. Justin is a PhD student at University of Albany, SUNY. Zach writes, hey, TWiM team, I love the podcast. Keep up the good work. Teresa writes, I'm sending this email and hope to be the lucky recipient of the 2-volume book, Manual of Clinical Microbiology. I'm a pediatric nurse practitioner practicing in a group practice in Southern California. I subscribe to TWiM, TWiV, and TWiP. I find all of these to be quite enjoyable and informative. Of note, I recently attended a pediatric conference last year, and one of the speakers was Michael Pickering, who's the director of Rochester General Hospital Research Institute. One of his lectures was It's in the Nose, Pathogenesis and Immunity. He presented research on the microbiome of the nasopharynx and the innate immune response there. I know more research from his department is ongoing, and hopefully you and your colleagues at TWiM will keep a lookout for research papers about this exciting research and its practical application to practice guidelines for care. Now I'm off to enjoy my sunny 58-degree weather and look forward to continued listening to your podcasts.
Nice.
Josh writes, greetings, TWiMers. I just listened to your most recent episode, and I want to stake my claim for the free book. Because I don't want to just send a blank email with only a subject, I just want to say that I've loved your show. I've been a dedicated listener for about a year now. I'm a graduate student in biochemistry, cellular, and molecular biology program at Johns Hopkins, currently rotating in Erin Goley's lab, which studies the biochemistry of FTSE, FTSZ. How about that?
It's another
cytoskeleton protein, right? I love your TWiX series for all the interesting papers you bring up. They occasionally provide really cool papers for me to do in Journal Club. Thanks for being awesome. One more from Juan. Dear TWiMers, I'm here to try my luck in the contest. There probably hasn't been 19 emails yet, but I missed the past few contests on the other TWi podcasts, so I'd rather send in this email sooner than later. Thanks for hosting this really engaging podcast. It has taught me a lot of microbiology while keeping my mind engaged working in the lab. Looking forward to an exciting TWi year ahead. And Juan is from Singapore.
So which one was he in the emailers? Do you remember which one he came in as?
No, I don't, but he was not 19.
No.
I'm sorry.
I knew that.
Let me just see. I could tell you Juan. Actually, so Mitch was the winner. Juan was very early on. He wasn't even close. But there have been 1, 2, 3, 4, 5 after Mitch. And, of course, this episode won't be released for a week, so we'll probably have many more. But keep on trying, folks. You know, I have a lot of books to give away. So today's is the Color Atlas of Medical Bacteriology, be the 12th emailer, subject line Color Atlas. And we love hearing about you and why you like the shows and so forth. So just let us know. And that'll be TWiM 144.
I don't get to play.
You can play.
Not fair.
You don't need books, do you?
I don't need books. I got enough.
You got enough. You can find TWiM at iTunes. It's also at a new place. It's at asm.org/TWiM. ASM has made a new website, asm.org/TWiM. Easy to get there. and also over at microbe.tv. And you should subscribe. Most people listen on their cell phone or tablet, and they have podcast applications where you can immediately subscribe to the podcast. Think about becoming a patron of TWiM and all of our other shows. Go over to microbe.tv/contribute. Have a variety of ways that you can help us through Patreon and other ways. And why do we want to do this? We'd like to travel a little bit and bring more of the crew with us. I often go places and do TWiVs or TWiMs and other podcasts on my own. I'd like to bring some of my co-hosts. And so if you support us, we can do that. If you want to enter the book contests or just send us questions or comments, the email is TWiM, TWiM, at microbe.tv. Michele Swanson is at the University of Michigan. Thank you, Michele.
Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
Make sure you do your laundry there, okay?
I'm going to make sure I do my laundry.
Elio Schaechter is at Small Things Considered. Thank you, Elio.
Hey, this was a blast.
Always. I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Chris Condayan and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. We thank Blue Apron for their support of TWiM. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.